Welcome back, students, to one more session of about soil microbiology. And today now we're going to deal with about soil protozoans. Soil protozoans, they are not an important member of the soil compared to the previously discussed members. Like you have bacteria and fungi, which have been found to be very large in number. But in the case of soil protozoans, they are being found to be less in number and they are not much of importance in the soil as compared to the bacteria and the fungi. These soil protozoans are usually found to be microscopic and their cells are being found to be devoid of chlorophyll. And they could be they could exist in the soil by doing either of two functions. They could be either be found to be saprobic feeders, that is, the decaying matter feeders, as well as at the same time they could be phagotropic. Now, what is phagotropic? Those organisms which could engulf the microbial cells directly are referred to as to be phagotropic. So the soil protozoans can either live in the soil as saprobic feeders, feeding onto the decaying matters, or they could be engulfing other microbial cells such as Enterobacter, Agrobacterium, Bacillus, etc. And if they engulf the microbial cells and grow on them, they are referred to as phagotropic. And you can find that the primary food of protozoans is being found to be bacteria or any other organic matter, as we mentioned. When conditions of unfavorable situations come, that is when the food has been diminishing or all that, these protozoans will form cysts, or they enter into some thick walls and they will form cysts, and they will remain inactive. And when the conditions become favorable, they will come out of the ends of the cyst and they will start dividing. So I guess you got an idea of what is a soil protozoan. Either it can be a saprobic feeder or it can be a phagotropic feeder. And under unfavorable conditions, it will go into form various conditions called encysted stages. And hope you do remember situations in which the soil bacteria are being hiding in between the soil pores such that they are being prevented from being engulfed by the protozoans. Now, if you go to look into the soil protozoans, they can be of different types. Sometimes they can have cilia on their body, or sometimes they can have flagella on the body, or sometimes they can have an amoeboid movement. So based upon the locomotion which has been possible for these soil protozoans, they can be either included under ciliata, they could be masticophora, or they could be amoeba-like movements. But unlike the other common protozoans, like we have learned about amoeba, paramecium, euglena and all that in our examples in the +2, but in the case of soil protozoans, the species of protozoans which have been found in the soil would be different from the common ones. And one example of a ciliata is Colopoda, and Masticophora, which has a flagella, example is Zercopoda. And you can find amoeba-like movement. You can find Euglypha. You can find you can find some hair-like structures over here, but at the same time it is also having a amoeba-like movement. So I guess the soil protozoans are clear to you. Now let's move on to viruses. Viruses, as we all know, they need some biological entity or a biological synthetic machinery of another host for it to survive. And the biological host of a virus could either be a bacteria, fungi, algae, actinomycete, and sometimes even certain nematodes. Okay, so if they are being found to be as viruses of bacteria, they are referred to as a bacteriophages. Okay, like that, either they are being found occupying the fungi, algae, actinomycetes and all that. And you can see that various nitrogen fixing bacteria like Rhizobium, Agrobacterium, Nitrobacter and all that, in them the viruses occur as phages. And some of the disease-causing agents of various agronomic and horticultural crops include various viruses. You will be learning about the different virally caused diseases in the coming sessions. I'm just not giving too much on it right now because we will be learning it in detail. And you can also see that you can in the picture which has been given here, you can see about the tobacco mosaic virus, which was growing on the tobacco leaves, okay, and which was the first virus which was being discovered, okay. And another way by which the virus can enter into the soil is sometimes you can see that viruses such as of the hepatitis virus can leak from the septic tanks, and they can move through the soil to the wells or other underground water supplies when leakage of the septic tanks do happen. So those soil, we consider it to be as free from much of viruses, but it can cause various pathogenic agents, pathogenic diseases. They can be serve as pathogenic disease causing agents, either in the case of plants as well as in the case of humans also. And most of the viruses, they remain active in the moist soil for a long time, and they get absorbed onto the clay particles or the humus. You know what is humus? The humus is a part, the inorganic material which is being left after. After it is resistant to degradation, okay. Like after organic particle degradation, there will be something left over which is cannot be degraded, and that is referred to as a humus. So viruses, as we all know, they will require a host, and they could be either pathogenic to the plants as well as the humans when they come across in the soil. And with this we come to an end of the viruses. And I would also like to talk about a small talk on the microbial loop. Okay. Now, what is a microbial loop? Microbes play an important role in the transfer of organic matter in different systems. Now, the degradation of organic matter can happen both in the terrestrial as well as the aquatic ecosystems. But it has been found that in the aquatic systems, the microbes are the most predominant members which will cause the degradation of the organic matter. But in the case of soil ecosystems, you can see that the major contributor in the primary production of organic matter in the terrestrial ecosystems mainly is being carried out by the plants, and the microbes have only a secondary role in the primary production of organic matter in the terrestrial ecosystems. And but in the waters we can see that they play it is a microbe which is the most important carbon or the organic synthesizer or the primary producing agent in the water is the case of bacteria. But we can find that microbes in the soil, they will recycle the organic matter which has been derived from the plants and the animals, and in turn these microbes can also be eaten up by various other predatory viruses and also predatory bacteria such as Lysobacter and Myxococcus as well as protists, and hence there is a recycling of the organic matter in the soil ecosystem by the microbes. So you have the plants which produce the most amount of the carbohydrates or the organic matter in the soil, and the plants are being in turn degraded by the microbes, and the microbes are in turn degraded by either predatory bacteria or by viruses or by protozoans, and thus the organic matter is being available to various other trophic levels in the case of the terrestrial ecosystem. And then another difference which we come across in the soil is that all the degradatory enzymes which are being released by these bacteria or the microbes in the soil are being found to be they do not rapidly diffuse away, okay, because in the case of soil ecosystem, what happens, the presence of water which will dilute the enzyme is being found to be very much less compared to the aquatic ecosystem, and hence what happens, the property of degradation will be found to be higher. The degradation of organic matter will be higher in a soil ecosystem than compared to that of a aquatic ecosystem. So let me just conclude right now. We have the different microflora of the soil, which consists of bacteria, fungi, actinomyces, viruses, and protozoans and algae, and all of them play an important role in the nutrient recycling of organic matter, nutrient recycling as well as the degradation of organic matter within the soil ecosystem. And it is a combination of all these microflora which will help in the growth of the plants, as well as the degradation of the plants as such, and helps us to maintain the food chain. Thank you for now. Hope so this is clear to you. We will be coming in details of various other sections of uh, agriculture as well as agricultural microbiology in the coming sessions. Thank you for now.